Good morning. I think from my previous sessions, you will get a basic idea about drug proving according to Hahnemannian perception. Now, I would like to invite your attention to another phase of drug proving called modern advancement of drug proving. The present day drug proving strategies are dramatically changed. To eliminate false signs and symptoms while proving, modern provings have modified from its concepts and its entire process. Now, let's see the recent advancements in drug proving. Without acquiring the knowledge of recent advancement in proving, the study of drug proving will not be completed. Drug proving, now termed as homeopathic pathogenetic trial or otherwise known as HPT. To eliminate false signs and symptoms and subjective bias, proposed the double-blind method for the drug proving in healthy volunteers. Here, the proving master and the prover should not be aware about the name of drug which conducted for drug proving. HPT is the first systematic experimental approach to detecting changes in the healthy volunteers according to the homeopathic pharmacopoeias in non-patient volunteers in relatively stable health conditions. Now, let's see about the current proving protocol. There are three basic components of homeopathic pathogenetic trials. They are the test substances, and the second one is the proving team, and the third one is the methodology. Now, let us see the test substances. The drug proving is conducted on healthy human being to note disease producing power. Here, drug is tested singly and alone according to the homeopathic pharmacopoeias. The test drug should be obtained from reliable pharmaceutical companies, and the detailed record should be kept of that company for future references. Now look into the test dose. The test drug should be proved singly and in pure and is in adulterated form. Details records regarding the source, collection, preservation of the drug substances should be maintained. The Latin name of the drug substances should be used wherever possible so that it can be accurately identified from similar sources, should be differentiated from other subspecies or some individual characteristics. Detailed descriptions about the preparations of the tested drug like maceration or percolation, succussion or trituration should be mentioned. Detail of vehicles and the scale of preparations of the tested drug should be mentioned. The type of succussion, that is whether the manual succussion or the automatic succussion is applied while manufacturing the tested drug, should also be mentioned. The proving is conducted with different range of potencies in centesimal, decimal and 50 millisimal scales, including mother tinctures, to get all aspects of the tested drugs. Before starting any proving, the physiological and the toxicological action of the drug should be studied in crude or material doses in lower animals. To avoid any bias, or influences on the part of volunteer or on the part of master brewers, the placebo is employed. The placebo, which should be the same color and texture as that of the tested drug, should be administered in the same way as that of the experimental group. Now, let us see the determination of the dosage. In drug proving, the drug may be used in crude form or low potency or in high potency depending on the nature of the drug. The drug, which even in its natural states affects the vital energy mildly, and those drugs proving only in high potency. Example, drugs like carbovegetabilis, graphitis and lycopodium, etc. In some other substances, their inert in natural state will not develop a proving, and such drugs need to be tested in only in potentized form. The drug which in the natural state disturb the vital energy to in a destructive manifestation should be proven only in potentized form. Example, medicines were taken from heavy metals, poisons and venom. And these substances are fatal in the natural states. Hence, the pharmacopoeia give cautionary details for dispensing the potencies of these remedies. That's all today. I will come again on tomorrow with the remaining portion. Until then, goodbye. Thank you.